loyalty, got royalty inside my DNA. Cocaine quarter piece, got war and peace inside my DNA. I got power, poison, pain, and joy inside my DNA. I got hustle, though, ambition, flow inside my DNA.
Hi, I'm Holly Byers.
And I'm Annie Byers.
And today I'm going to be asking her a few questions about nucleic acids. So Annie, what's a nucleic acid?
I don't know.
You don't know anything at all?
No.
The teachers at the junior high aren't teaching you anything about it?
No.
What an abomination. Well, hopefully we can help you find out.
Gabe Hartnett, what is a nucleic acid?
A phosphate group of a sugar.
Don't know? Okay.
Hi, I'm Katerina DeCamp with
Alexa DeCamp.
and today she's actually in the junior high. Today I'm gonna ask you a few questions on nucleic acids. Can you tell me anything about them?
No.
Really? Nothing?
No.
Do you know that they have something to do with DNA?
No.
Do you know what DNA
No.
is? You don't know what...
No. No.
Okay, well, stay tuned and you'll learn some more about DNA and nucleic acids.
All right, Nolan, former AP biology student. What is a nucleic acid?
A nucleic acid? It is an acid. I have no... Why are you asking me this? I'm not in this class.
No wonder he dropped.
Yeah. No wonder.
Hello, I'm Holly Byers here with Stephanie Fry outside of Marymount High School. And Stephanie, did you take freshman year biology?
I did.
So you should be able to tell me, what's a nucleic acid?
Can't tell you. Don't remember.
Does the word nucleotide ring a bell?
Not at all.
DNA? RNA?
I know DNA. A little bit.
So what do you think DNA is made out of?
Genetics.
I don't know.
The correct answer is coming up.
Jacob, what do you think a nucleic acid is?
Well, I'm kind of working on my DYOE right now, so can I postpone this interview?
All right, this AP bio student does not know what nucleic acid is. More later. Charlie Tripp, what is a nucleic acid?
It's an acid. I don't really know.
An acid?
Yeah.
All right, there you have it, folks.
Hi, I'm Katerina DeCamp and this is...
Daniela DeCamp.
She's with the junior high. Today, I'm gonna ask you some questions regarding nucleic acids.
Okay.
Can you tell me anything about them?
No!
Do you know that they have something to do with DNA inside your body?
No!
Do you know what DNA is?
No!
I'm teaching you a lot at junior high. Okay. Well, stay tuned for more about nucleic acids and DNA.
All right, Ian Mikesell, what do you think a nucleic acid is?
Nucleic acid is a macromolecule of nucleotides. It is a part of DNA, and may I say it consists of a sugar, and a phosphate group.
Wow, that is the best answer we have heard all day.
Thank you.
Congratulations, sir.
Thank you.
Oh my gosh.
Hello, Holly Byers here, reporting outside Marymount High School with
Emma Kuwach.
In the Reading Garden. Today, I'm going to ask you a few questions.
Yeah.
So, what do you know about nucleic acids?
I don't know. I don't know anything.
At all?
Nope.
Can you tell me something about nucleotides?
Some questions as her.
So are you aware that nucleotides make up nucleic acids?
Well, I am now.
I'm glad I could inform you. Could you tell me anything about how nucleotides and nucleic acids have a role in DNA or RNA?
Part of it. No, I don't know.
Well, I'm hoping that we can enlighten you on this.
Sounds good.
1 second.
All right, Jackson Comer. the news will not wait for anybody. So, what do you think a nucleic acid is?
Well, Lewis, I think a nucleic acid is something you put on a protein or as a functional group so that you can help make different proteins.
All right, folks, it looks like we're truly learning how much or how little Marymount High School students know about nucleic acids. So it looks like we're gonna have to teach them. Holly?
Hello, Holly Byers here today to talk to you about the macromolecule nucleic acids. So nucleic acids exist as polymers known as polynucleotides. And the polynucleotides are made up of monomers known as nucleotides. Now, what the heck does that mean, you're probably asking? I'm about to tell you. Alright guys, so here's my beautiful illustration that I've used to help you better understand what a nucleic acid is. So, as I said before, as I'm sure you all remember, a polynucleotide is the polymer that nucleic acid exists as. So it's a bunch of repeating nucleotides bonded together. So to make that a little easier to understand, I've drawn a single nucleotide so you can kind of get a close-up idea of what's happening. So a nucleotide always consists of three things: a phosphate group, wow, beautiful, a pentose sugar, super cute, and a nitrogenous base. And the nitrogenous base and the pentose sugar are part of a nucleoside, which is just the nucleotide without the phosphate group. So, the nucleotide, as I said, has three different parts: the phosphate group, the pentose sugar, and the nitrogenous base. But there are two different options for the pentose sugar that you use in your nucleic acid. Wow! So you can either have deoxyribose or ribose. You're going to use deoxyribose when you are working with the nucleic acids in DNA. And then you're going to use ribose when you're working with the nucleic acids in RNA. So the only difference between the two is that, as you can tell by the name deoxyribose, deoxyribose lacks an oxygen atom on the second carbon on the ring of the sugar. So that's the only difference between the two. And then you also have a couple different options for your nitrogenous bases. So you can either have pyrimidines, which are 6-membered rings of carbon and nitrogen atoms. And there are 3 different types of those. You have guanine, thymine, and uracil. So when you're working with DNA, you're going to have thymine, but when you're working with RNA, you're going to have uracil. So there are going to be no nitrogenous bases of uracil in DNA, and vice versa with thymine. And then the second type of nitrogenous base you can have are purines, which are 6-membered rings fused to 5-membered rings of carbon and nitrogen. So they're bigger than the pyrimidines. So you have adenine and guanine. Those are the 2 types of purines. And when you're making DNA and when you're like transcribing RNA, guanine pairs with cytosine and thymine pairs with adenine, and uracil pairs with adenine. So it just depends what adenine's going to pair with depending if you're in DNA or RNA. So I'm going to use my beautiful illustration once again to demonstrate how these nucleotides bond together to make this big macromolecule called a polynucleotide. So what happens is that through a process called dehydration synthesis, a water molecule is taken out to connect the phosphate group of one end to the 3' carbon end of a sugar of a different polynucleotide. So what ends up happening is there's a covalent bond between the phosphate and the 3 carbon end and the phosphate and the 5 carbon end of 2 different nucleotides, which is called a phosphate linkage. This ends up creating a sugar phosphate backbone that's repeating, but that does not include the nitrogenous bases. And just to give you an idea of what the two different types of nitrogenous groups look like, this is what is called a pyrimidine, or however you say it, and this is the pyrimidine. So as you can see, it's a lot bigger. Anyways, what this ends up creating is a 5' end and a 3' end. And this will become very significant later when we're talking about DNA replication and stuff like that, because you can only read from the 5' end to the 3' end. Anyways, the 5' end has a phosphate group bonded to the 5th carbon on the pentose sugar. But on the 3' end, you have a hydroxyl group bonded to a 3' carbon sugar of the pentose. So that's the difference, and that's just to give you guys a little background on the structure of nucleic acids. Take it away, Kat.
Whoa, thanks, Holly. Today, I'm going to be talking about DNA and RNA and their functions. So, there are two types of nucleic acids, and that is DNA and RNA. DNA and RNA allow genetic information to be passed from one generation to the next. DNA provides directions for its own replication, and it also directs RNA synthesis, which then controls protein synthesis. This whole process, that is called gene expression. Protein synthesis happens in the ribosomes. Eukaryote cells, this is in the cytoplasm, but DNA is still in the nucleus. Prokaryote cells lack nuclei, but they still use mRNA to convey a message from DNA to ribosomes, which then translate the coded information into long amino acid sequences. A given gene along a DNA molecule can direct synthesis of a tape RNA called messenger RNA. mRNA interacts with the cell's protein synthesizing machinery to direct production of a polypeptide which folds into a protein. The messenger RNA conveys genetic instructions for building proteins from nucleus to cytoplasm. So, as a whole, protein synthesis goes from DNA to RNA to protein. DNA also does not, this is a big misconception, DNA does not run the operations of the cell. So it's not doing the job, it just gives the people that are doing the job instructions for it. DNA is a genetic material inherited from parents, and each chromosome contains one long DNA molecule. When a cell reproduces itself during dividing, DNA molecules are copied and passed along one generation of cells to the next. So, going back to what Holly said, DNA molecules have two polynucleotides forming a double helix. In base pairing, adenine pairs with thymine, guanine pairs with cytosine. Going back to what Holly said, DNA molecules have two polynucleotides forming a double helix. In base pairing, adenine pairs with thymine, guanine pairs with cytosine. That's in DNA. And because these two strands, they're said to be complementary, this makes it possible to generate two identical copies of each DNA in a cell that is about to divide. Therefore, the structure of the DNA accounts for the function for transmitting genetic information. Linear sequences of nucleotides in DNA are passed from parents to offspring, determining the amino acid sequences of proteins. As a result, siblings have greater similarity in their DNA and proteins than do unrelated individuals of that same species. So there's actually a lot of similarities between two organisms that look totally different, such as humans and gorillas. They only differ in just one amino acid in their whole amino acid sequence, just one. And that creates such a difference between me and a gorilla. And also, another example is humans and frogs. They differ in 67, which of course is a lot higher number than just one. So that's why you get from me to a frog. So 67 differences in the amino acids. So to wrap things up from the new information that I just told you, you go to get from DNA or to get into a protein, you're gonna have to go from DNA to RNA to protein, and this happens in the protein synthesis happens in the ribosomes, which is in the cytoplasm for eukaryote cells, and the DNA is still in the nucleus. For prokaryotes, obviously, there is no nucleus, but they still use mRNA to get the message from DNA to ribosomes. Yes, thank you. Take it away, Lewis.
Katrina. Today, I'm going to be talking to you about how we see nucleic acids today. So, let's jump right into it. The Human Genome Project is a prime example of how nucleic acids play a large part in our lives. Nucleic acids are essential to life. They appear in all things that are living, and personally, I find that spectacular. So, the Human Genome Project was an international research effort to determine the sequence of the human genome and identify the genes that it contains and how nucleic acids and DNA affects us. Contributors to the project included universities across the United States and international partners such as England, France, Germany, Japan, and China. It's pretty amazing how the whole world came together just for this common cause. The Human Genome Project began in 1990 and was completed in 2003, 2 whole years ahead of its original schedule. That just goes to show how eager these scientists were to get to their very important work. So, the work of the Human Genome Project has allowed researchers to begin to understand the blueprint for building a person and how our nucleic acids and DNA impacts us. As researchers learn more about the functions of genes and proteins, this knowledge will have a major impact in the fields of medicine, biotechnology, and all of the life sciences. Well, so you've been watching this video, and I just know you are all wondering, how do I get more nucleic acids in my life since they are so awesome? Well, it's easier than you thought. Real-world nucleic acids are contained in fish, beans, legumes, and fruits, so it's as easy as eating an apple. Alright, we are going to jump into a little bit of a history lesson here. In 1869, some dude named Friedrich Miescher discovered nuclein, which would later be called nucleic acids. In 1889, my boy Richard Altman discovered that nuclein had acidic properties, so it became known as nucleic acid, the name that we all know and love today. Then, of course, we all know about the goats in 1953, Watson and Crick, who determined the structure of DNA. Great stuff there. Well, kids, I hope you learned something today. It is late at night on Sunday. I'm outside, so for those reasons, your favorite reporters, Holly Byers, Katrina DeCamp, and Lewis Gatch, signing off.